Hi everyone, in this video we'll be seeing about the hemoglobin, their structure and their functions. So under hemoglobin, based on their structure, it can be either deoxyhemoglobin or it can be either oxyhemoglobin. So why is it we have to study this? Of course, it is a very important question either in BSc or in MSc. Another reason to study this is if you want to know how we breathe in oxygen and how we breathe out CO2, then this is the video for you, because it will deeply explain our cellular respiration process. So hemoglobin main function is to transport oxygen. This hemoglobin is present in our blood, so that this blood contains oxygen in it. So first we'll see what is cellular respiration. Respiration means we breathe in oxygen. With this oxygen binds to the glucose molecule present in our body. So glucose, what is glucose? What we eat, especially we eat rice, right? So rice contains carbohydrates, that is carbohydrates is made of glucose. So this oxygen binds with the glucose and then it will release carbon dioxide, water and energy. So in order for our body to get energy, we need oxygen. Okay. How is this oxygen transported inside our body? Means it's because of this hemoglobin. So we'll see about some certain terms before going into the entire process. That is arterial blood. Arterial blood is nothing but oxygenated blood that is present in lungs, heart, in the left chamber and in arteries. So another is venous blood. So venous blood is nothing but it is deoxygenated blood. And another thing is alveoli. So this alveoli is present in our lungs. It is like a balloon, okay, where if we give the air to balloon, it will expand. If the air is released, it will shrink, right? Similarly, in this alveoli, when we breathe in, it takes in oxygen and it will expand. Once we breathe out, this alveoli will breathe out the carbon dioxide. So we will see the entire process. Initially, this hemoglobin will bind with the oxygen that is present in our lungs, and then it will carry this oxygen via the arterial blood to the muscles and tissue in our body. Where it transfers this oxygen means it transfers it to the myoglobin. So we are not going in detail about this myoglobin protein. Maybe in later videos we will be seeing. In myoglobin, the main function is it will store the oxygen. Hemoglobin will transport oxygen, where myoglobin will store the oxygen unless it is needed to produce energy from glucose. So once the oxygen is transferred from hemoglobin to myoglobin, later carbon dioxide, H2O and energy will be released. So consider if we are pregnant, then what happens is this hemoglobin oxygen is transferred to the fetal hemoglobin in the uterus of the pregnant mammals. So what happens next? Now CO2 is released and we have to get away the CO2 from our body. So again, this part is done by our hemoglobin, which uses the amino acids. So hemoglobin is made up of protein and heme group. We'll see in the next slide regarding the structure. So this protein is made up of many amino acids, and this amino acid present in hemoglobin will bind our CO2 and carry it via the venous blood back to the lungs. Once the carbon dioxide is taken by the hemoglobin to the lungs, in the lungs there is alveoli, which is containing the oxygen. Okay, because when we breathe in, it is taking now oxygen. Gas exchange reaction is happening between two. This alveoli will give this oxygen to hemoglobin, and this hemoglobin will give the CO2 to the alveoli. Okay, so that this alveoli is now having CO2 in it, and then this CO2 is released by the alveoli via mouth, nose. So this is the cell respiration process. Hope you liked this process. I liked it very much. So we have to know deep about hemoglobin, how it binds oxygen and how it releases oxygen and everything. So before going into that, we'll see the general structure of hemoglobin. It has two parts and four subunits. So four subunits, here it is mentioned in blue, red, violet and green. These are the four subunits. And as you can see, each subunit has two parts. One is the heme part and another is the globin protein part. So what is the heme part? If you have watched my earlier video, I have explained in detail about the heme group. So this is how the heme group will look. It contains a Fe metal, and this is the porphyrin structure. It is a metalloporphyrin and it contains around 8 substituents. So this is the heme group. So in every subunit there is one heme group. So there is around 4 Fe metal in our hemoglobin. Then we know that protein is made up of amino acids, and if you have learned the structure of protein, they may be in helix form. Helix is nothing but like a spring, okay, spiral. So it may be alpha or in beta form. So here two of them are in their alpha state and two of them are in their beta state, that is two of the proteins. So if the protein is in alpha state, then it contains 141 amino acids, and if it is in beta state, it contains 146 amino acids. Then each protein has one polar group and one non-polar group. So consider like a shell where the outer surface is covered by the polar groups and the inner is covered by the non-polar groups. So the outer surface is like hydrophilic and it is not water resistant, but the inner shell is protected from water. The inner cavity is fully hydrophobic because there are fully non-polar groups. That is, non-polar groups means non-polar groups that are present in the amino acids is present in the inner, but in the outer it is fully covered with polar groups. Then how is this four subunits bound with each other? So there is this NH3 plus and COO minus present in the protein chain. So if this blue color one has the NH3 plus and this red color one has the COO minus, then they both will bind. Similarly, there will be many bonds. This is like a salt bridge that binds all the four subunits. Then we'll see in detail about deoxyhemoglobin. So I hope this is interesting for you. Deoxy, as the name says, there is no oxygen. So this is the structure. You can see there is this is one subunit. Here it contains the Fe2 plus porphyrin structure, and this is the protein. Here in this proximal site, so this site is called proximal, that is below the ring, it is called proximal, and if something is attached above the ring, then it is called in distal site. Okay, since this histidine group, so this is the histidine group and this is the imidazole part that is in histidine group, and this nitrogen will bind to our Fe2 plus. So here this Fe2 plus is high spin and it is if it is Fe2 plus, then it is called ferrous, and it has a D6 configuration. Why is it having D6 configuration? Because Fe has an electronic configuration of argon 3d6 4s2. Once it releases 2 electrons, it will become argon 3d6. Okay. So there is just a fifth site occupied by histidine, and as you can see, this is not planar, and it has something like a pyramidal structure, like we can say planar pyramidal structure. Here they have mentioned in books like it has a square based pyramidal coordination because it is not planar. Then this is the electronic arrangement for this structure. So here we can see they are in high spin. Since it contains 6 electrons, it will be like 1, 2, 3, 4, 5 and 6. What is high spin means? There won't be any pairing. Okay, that's called high spin. Then another thing we can notice is that it contains 1 electron here in this position, that is in dx square minus y square, so that the electrons present there and the electrons that are present in the nitrogen, that is in the porphyrin structure, the electrons present in nitrogen and the electrons that are present in dx square minus y square, there is a repulsion between both of them. So due to this repulsion, what happens is the structure of the Fe atom will be a little bit larger, so it won't fit into the hole of the porphyrin, that is, it won't fit in this hole that is provided by the porphyrin ring. Rather, it will be a little bit above the ring, that is out of the plane of the porphyrin ring. So the ion lies like 0.36 to 0.4 Armstrong out of the plane. This is due to electron-electron repulsion. Then due to it containing unpaired electron, it is paramagnetic and it is red-purple in color. Then another thing is we know that proteins are bound together by, that is, the subunits are bound together by the salt bridges. Here all the bonds, that is all the salt bridges are very, very strong, so that there is more of steric hindrance, because since everything is a large molecule, there is more of steric hindrance, so there is more strain in the molecule. So that's why this deoxy state is called the tense state or the T state. Then coming to the oxyhemoglobin. In the case of oxyhemoglobin, initially this is the structure of deoxy form, and it has an Fe in Fe2 plus, and is of course we know high spin. And once this molecular oxygen, that is O2, binds here, it is converting into oxy form, where Fe is in +3 oxidation state and the OO has like this is in superoxide form. See, so what is superoxide means? O2 minus. So this is the initial form. We know the valence electron of oxygen is 6. You can count it 1, 2, 3, 4, 5, 6. And 1, 2, 3, 4 and 5, 6. So this is for O2 molecular oxygen when the Fe is in +2 state. But when Fe is in +3 state, the O2 looks like this, that is superoxide, O2 minus, so 1 electron is extra, so there must be 13 electrons. So 1, 2, 3, 4, 5, 6, then 7, 8, 9, 10, 11, 12, 13. So there are 13 electrons. So the structure is like this, O, O and then 1 minus. So this electron is allowed, it is not paired, as you can see, this electron, and the electron, 1 electron will be donated by this Fe2 plus. So that only it is converting into Fe3 plus, right? So that electron and the electron that is allowed in oxygen will make up this bond. Hope you are clear. So this oxygen is bound in the distal site. As I have earlier mentioned, this histidine is bound in the proximal site and this oxygen is bound in the distal site. So this oxygen is weak, but even though they are weak, they cause pairing, okay, like this. So since it is Fe3 plus, the configuration is argon 3d5 and their configuration looks like this. It is causing pairing. So 1, 2, 3, 4 and 5. So there is pairing as you can see. Then as you can see, this dx square minus y square, there is no electrons present here. So earlier I have mentioned that due to that electron, the nitrogen atom electron, there is repulsion. But in this case, since there is no electron present here, there is no repulsion between the electrons with the nitrogen atom. So since there is no electron-electron repulsion, the Fe2 is smaller in size and then it fits into the hole provided by the porphyrin ring very well. And as of now, you can see the overall geometry to be octahedral because 4 bonds in porphyrin and above and below we have 2 bonds, so totally 6 bonds. Then this entire structure is gives hemoglobin red color. So how do you know that when oxygen is binding to our hemoglobin, this oxygen is in superoxide form? This is confirmed using resonance Raman spectra. Generally, we know that if the strength of the bond is high, the distance between the bond is low. Consider, for example, you consider a single bond and a double bond. The double bond is stronger than single bond and the double bond distance between the two atoms is very low than single bond. Similarly, if the bond is strong, then the stretching band frequency will be high. You might have learned in IR. Here also it's the same case. So these are some fixed values that are taken in resonance Raman spectra for various O2 species. Here you can see that O2 plus, after it has donated an electron, their bond is very strong. That's why it has a very high stretching band frequency. And as you can see, the distances between the O bond is very low compared to others. But you can see in O2 2 minus, it is having very low stretching frequency because the bond strength is very low and the distance between the two O is very, very large. So from this we can conclude, how we can conclude that superoxide is the thing means, they have calculated this resonance Raman spectra for oxyhemoglobin and they have found that the range is 1105. This 1105 matched well with 1097, that is superoxide. That's why they have concluded that O2 minus may be the form of oxygen that is present in oxyhemoglobin. Then how is this oxygen binding to our hemoglobin? The first oxygen will bind to one of the heme group. Okay. It is very difficult to bind the first oxygen in the entire hemoglobin. Once the first oxygen is bind to one of the heme group, the salt bridges will be broken between most of the subunits and there is also some conformational changes in the protein that is happening so that everything will go into a state called relaxed state. There is no more tension because there is no more steric hindrance. So that later on the oxygen binding in other 3 subunits is very, very easy because there is no hindrance for binding of oxygen. So this effect is called cooperative effect. You can spell it out and see the phenomenon where the addition of dioxygen to one heme subunit encourages addition of dioxygen molecule to other heme subunits is called cooperative effect. Then this is the chemical formula for that. This is happening in lungs as I have shown in first video, that is first slide. The hemoglobin will bind with 4 oxygen because it contains 4 subunits and it will give hemoglobin O24. So that's it. And another thing you have to notice it, once the oxygen binds, the Fe, which was present initially above the ring, it will fit into the hole once the oxygen is bound, and also this histidine group will move nearer to our porphyrin ring. It won't be going away, it will move nearer to the our porphyrin ring. Another question you may ask, how is this O2 minus stable? This O minus will bond with the imidazole hydrogen, and there is hydrogen bonding. Of course, some other protein molecule in distant site will have imidazole in them. Since you know that this heme group is surrounded by protein, so that imidazole hydrogen, this is also imidazole. Some imidazole hydrogen like this will form hydrogen bond with this oxygen, so that they are stable. So how do we differentiate between hemoglobin oxygenated form and deoxygenated form? So this is HBr represents reduced hemoglobin. Why is it reduced? Means reduction is nothing but gain of hydrogen and loss of oxygen. So since there is no oxygen, it is called reduced hemoglobin. So this reduced hemoglobin is this dotted line. I hope you know this UV visible. It is measured in wavelength in x-axis, and here it is measured in molar extension coefficient y-axis, and this is the reduced one, that is the dotted one. But once it's oxygenated, it gets 2 bands, as you can see. So this is how we can differentiate easily between both forms. Hope you like this video. My main references are from Stephen Lippard Burke and Ajay Kumar. Ajay Kumar is my very favorite book. I think I have tried to explain every key points possible for all of the exams you want to learn. I hope you enjoyed this video by knowing about cellular respiration. If there is any doubts or queries, you can mention in comments. I'll answer it. I'll love to answer it. And then thank you for watching this video. Thank you.